Hi class, this is your instructor, Scholar Huff, and we're back here again, and this is now the 30th chapter, An Introduction to Animal Diversity. Although members of most animal species are readily recognizable as animals, the identity of some others are much less obvious. Early naturalists thought sponges were plants because they were anchored in one place. However, many people still make that mistake among certain marine animals, such as those sponges and corals, for plants. So if you look in your text on page 628, you will see just that they may not appear to be that animal that you know and, of course, easily recognize. So locomotion is not a requirement for being classified as an animal. So animal phylogeny is an exciting and rapidly changing field of study. Biologists have described and named more than 1.5 million species of animals. And then each year, an approximate 15 to 20,000, meaning 15,000 to 20,000 new species of animal are named each year. And millions more probably remain to be discovered and classified. Though, interestingly enough, that there has been an estimated 99% of all animal species that have ever inhabited our planet are now extinct. Keep that in mind, class, as we do continue. So with that being stated, just know that there are animals that are here and in existence. However, many of those animals are no longer here in existence today. So with that being the case, taxonomists have assigned extant animals to about 35 major groups or 35 major phyla, and molecular studies have confirmed that many of these groups are monophyletic. That is, that all descendants and only descendants have a common ancestor. So with this, that just takes you back to a bit of evolutionary terms and the relatedness of animals, meaning it concludes not just, of course, the most recent common ancestor, but it also includes all descendants of that most recent common ancestor class. So animal groups that are not monophyletic have been split or reorganized, with some members of the groups being reassigned to other taxa. So with that in mind, it is important to remember the classification of animals and the relative positions of animal groups are a work in progress as they consider new data. Systemicists redraw the tree of animal life. So in this chapter, we will discuss those characteristics of animals and their habitats. We will then explore animal origins and some of the criteria used to determine the evolutionary relationships to classify animals. And then we'll finally introduce the major animal groups, including the 3 major clades of bilateral animals. So do keep in mind that this is giving you that introduction to animal diversity, including, of course, how animals have been classified. So let's begin with the characteristics. You will see these on your test, and prepare yourselves to, of course, go through each 8 of these on the test. So animals share 8 characteristics, meaning 8 defining characteristics. They are as follows. The first one being class that animals are multicellular eukaryotes with cells lacking cell walls, unlike plants, algae, and fungi. So as animals are, they gain structure through collagen and other structural proteins. So just keep in mind that that structure does depend on that extracellular matrix, which of course our cells secrete. So collagen is that important, a majorly important shared derived character in animals. Next up, animals are heterotrophs, relying on producers for raw materials and energy. So animals ingest, then digest within the digestive system. So with that being the case, it's not the case that we can produce our energy, as of course plants do. Next up, cells comprise the body, and they are specialized to perform a specific function. So all in all but the simplest animals, cells are organized to form tissues. So keep in mind that it's the cell, not the tissue, that defines the animal. So in small animals with simple body plans and life processes such as gas exchange, circulation of materials, and waste disposal can take place by diffusion of gases and other substances directly from the environment. And of course, in us, such as larger animals, the specialized organ systems perform those functions. The fourth characteristic. Animals exhibit a diversity of body plans. So it's not that shared body plan that we as animals have. So its basic structure and function is by way of the design of the body. So animals have adapted to its method of obtaining food and reproducing. Up next, number 5. So animals are capable of movement or locomotion at some point during their life cycle. So with that in mind, sessile animals cannot escape those unfavorable conditions, such as you would see class in the case of those adult sponges. They are not allowed. They are not able class to move around. So the sixth characteristic. Most animals have a nervous and muscular system. And yes, I did say most. And they are capable of responding to environmental stimuli. Next up, the seventh characteristic. Most animals are diploid, and they reproduce sexually with large eggs and flagellate sperm. So, of course, that flagellate sperm, upon fertilization, of course, with those two cells, they then develop into what is known as that diploid zygote. And then, lastly, most animals go through that period of embryonic development. So, of course, by way of embryonic development, we have, of course, cleavage that occurs. And when I say cleavage, that refers to those mitotic divisions. So as those animals go through the mitotic divisions, of course, they will eventually develop into the blastula, which, of course, the blastula is going to be that ball of cells. And then most animals then further develop into what is known as a larva, that sexually immature form that may look different from an adult. And then the larva, yes, it differs from the adult in many ways, including where it lives in its habitat, how it moves, and even what it eats. And then the larva typically undergoes what is known as metamorphosis, be it complete or incomplete. So by way of metamorphosis, it then develops into the juvenile and ultimately class the adult. So there you have it. Those class are the 8 defining characteristics of animals. Please make sure you are able to go through those on the test. So next we get to the adaptations to, of course, life from the ocean to freshwater and terrestrial habitats. So let us begin there now. So with this in mind, as animals have developed, they, of course, all began class in the sea or in the ocean. So marine environments offer an abundance of advantages. So the buoyancy of seawater provides its support. And that large volume of water keeps the temperature relatively stable. Another thing that happens, of course, is that the body fluids of most invertebrates have about that same osmotic concentration to the seawater. So the fluid and salt balance are more easily maintained there than in fresh water. So plankton class is, of course, in abundance. So that mainly consists of the microscopic animals and protists that are suspended within the water. So it provides that readily available and abundant source of food for those aquatic animals. However, life in the ocean also does present challenges. Although the continuous motion of water brings nutrients to animals and washes their wastes away, animals must be able to cope with the water's movements and the currents to, of course, keep them in point A and not get to point B. So squids, fishes, and other marine animals have evolved as being strong swimmers, usually able to direct their movements and maintain the locomotion. However, most invertebrates and young vertebrates cannot swim strongly, and they have adapted in various ways to, of course, the tides and currents. So some sessile animals, which are permanently attached to a stable structure, such as rock. So other animals burrow into the sand and silt that cover the sea bottom. Then again, most invertebrates have adapted to maintain that small body size, becoming part of the plankton. So they are tossed about and the food supply continues to surround them. So please just keep in mind that plankton is abundant, buoyant support is provided, there are stable temperatures, and the body fluids class retain an osmotic pressure class similar to that of the sea. Next against fresh water. Next up is fresh water. So in freshwater, life is more difficult class than is in the ocean. So there are fewer types of animals that make their homes in freshwater than in the ocean because living here in this habitat is more difficult. Freshwater is hypotonic to tissue fluids of animals, so water tends to move into the body by osmosis. So to survive in this habitat, freshwater species must have mechanisms to remove excess water while retaining salts. By way of osmoregulation, I repeat, by way of osmoregulation class, this, of course, expends energy. So they must expend energy class to, of course, maintain the appropriate osmoregularity. So continuing here, freshwater offers a much less constant environment than seawater. So animals that inhabit fresh water must have adaptations for surviving variations in oxygen content, temperature, turbidity, and even the volume of water. And I did say turbidity. And when I refer to water being more turbid here, that would be that the sediments within the water are going to be what is called suspended. So maybe you've seen clear water before, but water that doesn't appear to be clear, such as water that's in a pond, water that's in a river, is more turbid because of those suspended solids within the water. And just to ensure class, yes, there is less food in fresh water, but of course there is, I repeat, no bearing on the number of animals in fresh water due to there being less food in the water compared to the ocean. So thirdly, class, we'll get to that terrestrial environment. So terrestrial living requires major evolutionary adaptations. So living on land is even more difficult than living in fresh water. And the evolution of terrestrial animals involved major adaptations. So analyzing the fossil record, many biologists hypothesized that the first air-breathing terrestrial animals were scorpion-like arthropods that came ashore during the Silurian period about 444 million years ago. The first vertebrates to inhabit terrestrial environments, the amphibians, did not appear until the Devonian period, about 30 million years later. So the most difficult aspect of terrestrial living class is desiccation, or drying out. So water is constantly lost by evaporation and is difficult to replace. So a body covering adapted to minimize fluid loss helps to solve this problem in many terrestrial animals. Location of the respiratory surface is deep within the animal, and that also helps to prevent water loss. Thus, the gills of aquatic animals are typically located externally or near the body surface. But lungs and tracheal tubes of terrestrial animals are typically found deep within inside the body. Reproduction on land also poses a challenge to protect the gametes and the developing offspring from desiccation. So aquatic animals typically share their gametes in the water where fertilization occurs. Some land animals, including most amphibians, return to the water for reproduction, and their larva forms develop in the water. So the evolution of internal fertilization has permitted many terrestrial animals, including land planarians, earthworms, land snails, insects, reptiles, and that does include birds, and terrestrial animals, transfer sperm from the body of the male directly into the body of the female by copulation. A watery medium continuously surrounds the sperm. Another important adaptation to reproduction on land is a tough, protective shell that surrounds eggs of many species. Secreted by the female, this shell protects the developing embryo from drying out. An alternative adaptation, however, for terrestrial reproduction is the development of the embryo within the moist body of the mother. So water has buoyancy that helps support animals that inhabit aquatic environments. Air is less dense in water, and to inhabit the land, animals must have structures such as a skeletal system and muscles that support the body. The temperature extremes of terrestrial habitats also present challenges. So just keep in mind that life on land is most difficult, namely because of desiccation. So they have the body covering with deep lungs within the body, reproduction occurs, of course, by way of that internal fertilization. There is embryonic development within the female. There is an egg with a shell, and there is a skeletal system class with muscle. Please prepare yourself to contrast, class, those evolutionary adaptations for life in the ocean, life in fresh water, and of course, life on land. This continuum that I just gave. So there you have it, class. So from here, we get directly into animal evolution. And this continues on class page 631, section 30.3. So with this being here, class, keep in mind that animals share a common ancestor with the protists. And what I'm getting to, class, is that the choanoflagellates, being that ancestor, including, of course, fungi and animals, are opicus. And as I say it that way, I just mean that we all share that choanoflagellate ancestor. So molecular analyses indicate that the RNA of all animals is very similar. So what I'm getting to is that we have parsimony. And when I refer to parsimony or us being parsimonious, I'm saying that it is unlikely that animals evolved multiple times, class. And that's majorly important for animal evolution. So, as I continue through here, we're now getting into what is known in class as that Cambrian explosion, or the Cambrian radiation. So, this was that rapid evolution of the amazing variety of these modern-day body plans. So, it occurred in approximate 500 million years ago, this Cambrian explosion did, and it was not an actual explosion, you all. So, as it happened, this radiation event is why we now see in existence most extant animal phyla. So at the beginning of that Cambrian explosion, there, yes, was a diversity of body plans, and what has now occurred is this is why we see what we now see, and we're about to get into, class, with these differing groups of animals. So with this, we're getting into animal phylogeny just a bit, and we'll get to Hox genes. And when I mention these Hox genes, I'm getting into, of course, them being found only in those bilateral and radial symmetrical animals. And as I get to this, what I'm getting to now, class, gets more so into being this, I guess I'll go to the next portion, about these hypotheses about the evolution of the development of animals. So yes, that basic set of genes has gotten us to where we are today, and of course, where we were with those changes in body plans, but it's all about those patterns seen by way of embryonic development. So biologists have long used those similarities and differences in embryonic development to hypothesize how the animal groups are related. So before and now, or at least traditionally, biologists depended on those main structural differences to compare the processes of development in various groups. Well, what we're getting to now, class, is this evo-devo, meaning that evolutionary development biology, meaning it's become an important approach to studying both animal relationships. So it's the comparison of those molecular events, such as gene regulation, during the process of development in those various groups. So let's get now, class, further into it. So as we reconstruct the animal phylogeny, what happens here, or what we're doing here, is biologists use those similarities and differences in morphology to infer those evolutionary relationships, such as symmetry, the number of tissue layers, referring to them having only 2 germ layers being diploblastic, or having 3 germ layers, class, being triploblastic, the presence of that body cavity, or the type of body cavity, and even the pattern of development. So using all of this, class, is how we get to where we are today with animal classification. So on your own time, class, please ensure that you all review these basic types of characteristics here, such as cephalization, meaning that development of the head region, or at least the head, where you have those sensory structures concentrated. And also, class, just review terms such as being anterior and posterior, dorsal, and even, class, ventral. And I mentioned tissues. We'll get more class into tissues in the 39th chapter, which is the next chapter, and the second of 2 chapters on this next test, and even germ layers, which is just mentioned class, being diploblastic or triploblastic. And even class review body cavities. Meaning I won't go through that at this moment, but you should review this because I'll get further into this class in the next chapter. As we get into the 31st and 32nd, of course, chapters. So from here, class, I'll continue myself on with those 2 main clades. Bingo. So what I'll get to now, class, begins for you all on page 635 of your text. And prepare yourselves to contrast these on your test. So with this, bilateral animals form 2 main clades based on differences in development. So I'm going back a bit, class, to those cells of the blastula. As they undergo gastrulation during cleavage of the embryo, we get these 2 main clades, the Protostomes class and Deuterostomes. You all will be tasked with listing, of course, phyla that are included in each of those 2 main clades. So listed for you, class, are them. So the Protostomes include class, phylum Mollusca, phylum Annelida, and phylum Arthropoda. The Deuterostomes class include the echinoderms, so of course, phylum Echinodermata, and the chordates in phylum class Chordata. So in your test, class, just prepare yourselves, and to help you all with this, make yourself a quick T-chart. So in said T-chart, you can place class Protostomes there on the left, and I'll abbreviate Protostomes as proto. You should write the entire word out, and I'll abbreviate class Deuterostomes as D-E-U, deuter. But of course, class, as you're writing out your notes right now, please write out those entire words, class, and let's now begin. So let's go to Protostomes first. So the Protostomes, class, I'm giving you the characteristics. They go through, class, what is known as spiral cleavage. So with spiral cleavage, class, I'm referring to those cells dividing, class. So they divide spirally, so you have, of course, 2 cells up, and of course, 2 cells down at that approximate, class 45-degree angle. That's how it occurs. Additionally, class, they exhibit what is known as having determinate cleavage. So since I mentioned it being, class, determinate cleavage, that does mean if they lose a cell, class, during development, they likely could very well lose a limb, class. Up next, class, getting to the first blastopore opening, it is, class, the mouth. So the mouth develops, class, before the anus. This is the Protostomes. In Deuterostomes, however, class, it is radial cleavage that occurs. By stating, class, it's radial cleavage, that means as these cells go through those mitotic divisions, class, these cells divide, class, directly above and below at 90-degree angles, as opposed to what we saw, class, with spiral cleavage in the Protostomes. So these animals exhibit, class, what is known as indeterminate cleavage. So if they lose, class, a cell by way of going through cleavage here, class, other cells then compensate. And in case that doesn't make much sense, class, this is what has led to developments, class, in vitro fertilization. In that, let's say this is a petri dish here. Petri dish. So in said petri dish, class, with appropriate media, you can insert, class, or at least have there, egg and sperm. So egg and sperm, class, fertilize. So from that fertilization event, class, you then have zygote. Well, this zygote here, I'll put 2 in, because that was in. Put them together, class, you get back to, of course, diploid being 2 in. That's a plus sign. So the cell, class, can then undergo cleavage, meaning in this medium, class, can, of course, continue to develop. After approximately 5 days, then a cell can be removed. And this cell, once removed, you can then take what was being developed in, class, and freeze it to arrest it, class, in development. So this cell that was removed, or at least that has been removed, can then be tested for an array, class, of genetic disorders. I would say hundreds of genetic disorders. And now I'm not saying class just one zygote would be class created, but there would be many of these, and of course many cells to test. So upon the test classes and determining which of those embryos class are whole with an exactly class 46 chromosomes, nothing more class and definitely nothing less, you can then class implant that frozen class, of course upon thawing, embryo into, of course, the mother or, of course, some surrogate. And it's all based on this being that we, and of course others, of course, animals, exhibit class indeterminate cleavage. And then lastly, class, the anus is that first blastospore opening. So the anus class develops before that mouth part. So with this, we'll further classify animals class, but we won't necessarily get into those two major clades in this respect. What we will do, however, class, we'll be dealing with what you see here. So on your next exam class, you'll be given the cladogram. And using the cladogram, what you all will add to said cladogram are those synapomorphies. And you say synapomorphies, correctamundo. And I'm just getting at, class, the synapomorphies are going to be the place at which there was evolution of segmentation, or the Deuterostome pattern of development, or even which the Protostome pattern of development, i.e., symmetry class being bilateral, i.e., evolution of tissues, i.e., class multicellularity, the definition class of animals. So keep in mind, class, we're now in animals, and of course, you're seeing it all beginning class there with that coanophyte ancestor. So please prepare well for your test class. This has been Chapter 30. If you have any questions, feel free to contact me, class, stop by my office, or even send an email. This has been your instructor, Scholar Huff, and thank you all for listening and prepare well.